Hello everyone. In this class, we'll try to learn about how carbon dioxide is transported in the blood. Carbon dioxide is produced mainly during aerobic cellular metabolism of glucose and during conversion of carbohydrate to fat. From tissue, carbon dioxide is transported to lungs in the venous blood and then exhaled in the expired air. The carbon dioxide produced by oxidative metabolism in the tissue, which increases the content of carbon dioxide in the cell. Carbon dioxide diffuses from the cell into tissue fluid and then from interstitial fluid into the tissue capillaries, which is then transported by blood to the lungs. In the lungs, it diffuses into the alveoli and then get expired to the atmosphere. Carbon dioxide can diffuse about 20 times as rapidly than oxygen. Therefore, the pressure difference required to cause carbon dioxide diffusion is much less compared to the pressure difference required for oxygen diffusion. Partial pressure of carbon dioxide in the cell is 46 mmHg. In the tissue fluid, it is 45 mmHg. Therefore, from the cell, carbon dioxide diffuses into the tissue fluid. The arterial blood that reaches capillary has PCO2 of 40 mmHg. Hence, carbon dioxide rapidly diffuses from tissue fluid into capillary blood. Two factors influence the diffusion of carbon dioxide from tissues into capillary blood. The first is the rate of tissue metabolism and the second is rate of tissue blood flow. A decrease in blood flow from normal to 1/4 normal increase tissue PCO2 from the normal value of 45 mmHg to 60 mm of Hg. And conversely, increasing blood flow to 6 times to the normal decrease the interstitial PCO2 from the normal value of 45 mmHg to 41 mmHg. An increase in tissue metabolism greatly increases the interstitial tissue fluid PCO2 irrespective of the rate of blood flow, whereas decrease in the metabolism causes fall in the interstitial fluid PCO2. So this showing the different levels of the PCO2 in the artery. So in the artery, the PCO2 is 40 mmHg. And compared to the PCO2 in cell, it is 46 mmHg. And by the end it comes to the venous blood, the PCO2 will be 45 millimeters of mercury. The modes of transport of carbon dioxide. Carbon dioxide is transported in three forms. One is physically dissolved in the plasma, that is 7%, as a bicarbonate ions in the plasma and in red cells 70%, as carbamino protein complexes 23%. So some of the carbamino compounds can be they can form complex with plasma proteins and carbamino hemoglobin in red cells. Dissolved form in plasma. The amount of carbon dioxide dissolved in the plasma is proportional to the partial pressure of carbon dioxide. As the principle of Henry's law, the solubility of coefficient of carbon dioxide is 20 times that of oxygen, that is it is around 0.06 ml per deciliter per mmHg. Thus, in venous blood, 2.26 ml per deciliter, that is 46 mm of Hg PCO2 in venous blood into 0.06 is carried in simple solution. Due to the absence of carbonic anhydrous in plasma, the carbonic acid formation is less in plasma. However, small amount of carbon dioxide combines with plasma proteins as the carbamino protein complexes. As bicarbonate ions, about 70% of the carbon dioxide is transported in the form of bicarbonate ions in the red cells. This becomes possible due to the presence of large quantity of carbonic anhydrous in red cells. From tissues, as soon as carbon dioxide is released into the plasma, a bulk of it diffuses into the red cell. In red cells, it forms either carbonic acid or carbamino hemoglobin. Carbon dioxide combines with water to form carbonic acid by carbonic anhydrous. Then carbonic acid spontaneously converted into H plus ions and bicarbonate. Okay. So here you can see there will be formation of H plus ion and bicarbonate ions. As carbonic acid readily dissociate into HCO3 and H plus ions, bicarbonate concentration increases in red cells. HCO3 leaves the red cells and in exchange to chloride which enters the cell from the plasma to maintain electrical neutrality. The exchange of bicarbonate for chloride occurs by chloride bicarbonate exchanger, that is an anion exchanger present in the red cell membrane. This exchanger is an anion exchanger AE1, which was previously known as band 3 exchanger. The chloride movement in exchange for HCO3 is known as chloride shift. As this phenomena was described by Hamburger, chloride shift is also known as Hamburger shift. So here you can see the carbon dioxide which diffuses into the carbon in the red blood cells in presence of carbonic anhydrate gets converted into bicarbonate and H plus ion, and excess excess bicarbonate ions get exchanged with band 3 protein. Okay, and for which the chloride is going to enter inside. So the H plus ions thus formed, they are going to combine with the oxygenated hemoglobin and they're going to release the oxygen, and the oxygen is released to the tissue. The H plus formed during reaction cannot easily diffuse out of the cell because the low permeability of the cell membrane to H plus ions. Most of the H plus ion produced by the dissociation of bicarbonate is buffered by hemoglobin. As H plus binds to hemoglobin, it decreases the oxygen binding of Hb and shifts the oxygen dissociation curve to the right. This facilitates the release of oxygen from hemoglobin into the tissue. This also favors transport of carbon dioxide. Reverse process operates in pulmonary capillaries, whereas the oxygenation of hemoglobin favors the unloading of carbon dioxide. As a carbamino compound, the carbon dioxide in red cell also reacts with the free amino groups of hemoglobin to form carbamino hemoglobin. And deoxygenated hemoglobin binds more carbon dioxide than oxygenated hemoglobin. About 23% of carbon dioxide is transported in this form. Carbon dioxide dissociation curve. Carbon dioxide dissociation curve is plotted in a similar fashion to that of oxygen. Unlike oxygen dissociation of carbon dioxide from blood is directly related to the PCO2. Therefore, carbon dioxide dissociation curve is almost straight line in normal arterial PCO2 range. So this is when you plot carbon dioxide dissociation curve at the pressure of PO2 40 mmHg and at the pressure of 100 mmHg. Okay, so in presence of the oxygen, the carbon dioxide dissociation curve is going to shift to the right side, leading to deloading of the carbon dioxide at the tissue, that is at the lung. The major features of carbon dioxide curve are the linear relationship between carbon dioxide content and PCO2. Within the physiological range of PCO2, that is between 40 to 46 mmHg, the curve is more linear and steeper, whereas oxyhemoglobin dissociation curve is neither linear nor steeper within the physiological limits of PCO2 from 40 to 100 mmHg. Transfer of large quantity of carbon dioxide can occur because of the shape of the carbon dioxide dissociation curve. Okay. So large amounts of carbon dioxide can be loaded or unloaded from the blood within small change in the PCO2. Also, blood can hold much of carbon dioxide than oxygen. The carbon dioxide dissociation curve shifts downward and to the right at a higher PO2. The effect of change in PO2 on carbon dioxide content is known as Halden's effect. The relationship between PCO2 and carbon dioxide content is inverse. At any level of PCO2, the total carbon dioxide content increases as PO2 falls. The advantage of this inverse relationship, that is the Halden's effect, is that it allows the blood to load more carbon dioxide in the tissues and unload more carbon dioxide in the lungs. Thus, when blood enters capillaries in the tissues and releases oxygen, the carbon dioxide carrying capacity increases, and conversely, when blood enters pulmonary capillaries and picks up the carbon dioxide carrying capacity decreases. Halden's effect. When blood passes through pulmonary capillaries, oxygen diffuses into the blood and forms oxyhemoglobin. Oxygenation of hemoglobin shifts the carbon dioxide dissociation curve to the right, and HP begins to lose carbon dioxide. So thus in lungs, loading of oxygen facilitates unloading of carbon dioxide. So this is called as Halden's effect, as it is named after great ancient respiratory physiologist John Scott Halden. As deoxyhemoglobin binds H plus more actively than that of oxyhemoglobin, binding of oxygen with hemoglobin decreases its affinity for carbon dioxide. So that is in brief about the carbon dioxide, the transport of carbon dioxide and the carbon dioxide hemoglobin dissociation curve. Thank you.